I'm going to try and do something a little bit different today. I'm going to not be using the iPad to draw on. I'm going to be using it to film the screen right here. So, hello. My hope for this is that the demonstration that I'm going to use, you'll be able to watch. We're not going to be exactly taking notes along with what's going on because we're talking about glycogen breakdown today. That's going to be our main topic. Most of what we go over is going to be right there. So that's going to be our main portion for today, and we're going to do a little arts and crafts to help describe it. And I think moving the little pieces of paper around and seeing what moves where is going to help you guys kind of conceptualize what's occurring during glycogen breakdown just a little bit more. First, though, item number 5 is the homework that I talked about last class. Some of you may have seen in the hour before class I posted an update on that. I've pushed that date back to Monday when that is due. If you scrambled to get it done for today, congratulations, you're done. You can relax and wait till Monday to hand it in. Also, Brandon came to my office and said, you know, I can't find TBC1D1. I don't know what the heck this is. The two of us looked through a number of research articles for about 5 minutes. And I said, you know what, if I can't find it in 5 minutes, this is hard to find. So you'll get bonus credit if you are able to find the full name of that. Not just TBC1D1, what it actually stands for. What's the T stand for? What's the B stand for? D1D1. Yes?
Say that we found that, but it's like the abbreviations of things.
I need the whole thing. Exactly. Yeah, the abbreviations are there. But I need to find out what those abbreviations stand for. So the T may stand for tray or something like that. What's that tray mean? Okay? That's what I need to find out. Other questions about the homework? Print that out, bring it in. Those abbreviations need to be used and explained along with the steps. So I want you to rewrite the steps that we talked about for both insulin-mediated and contraction-mediated glucose uptake. Okay? Just a little bit of repetition is going to help you guys understand that a little bit more. So we're going to do a little bit of glycolysis regulation, and just if we have time, we'll do some lactate shuttling as well. So, last class we talked about glycogen. We talked about the difference between our bonds, and we have 2 main different bonds. What are those? What are the 2 types of bonds? Alpha 1,4, alpha 1,6. And that's going to get really important when we start talking about the different enzymes that break down glycogen. And overall, what's our purpose? Why do we want to break down glycogen? Energy, that's very broad, that's a very philosophical answer. Let's be a little bit more specific. Dan, what do you have?
To get to the glucose.
To get to the glucose, to get to the what? To get to the ATP, first you get the glucose, then you get the respect, then you get the ATP. That was in a movie I think, Scarface, right? No, but we want to get to ATP exactly, but the way we get there is through glycolysis. All right, so we need to get glycogen into glycolysis. So we need to break down these. Remember, this guy right here is our branch. So this is our alpha 1,6, and up here this is our alpha 1,4. We understand why we have a lot of N-terminals, and the reason is because it helps break down glycogen more quickly. Let's just do some quick definitions because these will help us define what we're going to talk about today. So glycogenesis is the formation of glycogen. On the other side, glycogenolysis refers to glycogen breakdown, and the reason we know that is because it's got the suffix on the end there, lysis, that means to break. Generally, you're going to see that in forms of lysing cells, and that's going to be when you break apart the cell membrane and spill out the inside of that. But as far as this is concerned, it's the breakdown of glycogen. Okay, so how does glycogen get broken down? So let's start. What is glycogen made up of in the first place? Allie, what do you got? What do we put together to make glycogen? Thank you, Allie. Glucose. Okay, so here we go. We got a bunch of glucose. Oh, let's see if we can squash these in a little bit more here so we can get all 5 of them. It's important that we get 5. Okay, so you see we have 5 different glucose, but remember we said that glycogen has a lot of branches in it. Those branches help us be able to break it down faster, so let's put a branch here and some more glucose. So we're going to do this just like so. Oops. Let's move all these guys up to the very top. So we got room. So here's our branch. Just like that. And a couple more glucose. Now, we talked about our bonds. Here's our alpha 1, 6. Can you see that? Oh, you can't with the stupid record button right there. There we go. Alpha 1, 6. And then between the rest of them, we have our alpha 1, 4s. Okay, alpha 1, 4, alpha 1, 4. Why do we call them 1, 4, and 1, 6? Here's some rumblings. Yeah, the carbons are numbered, so it's which carbons are being connected. So it's the carbon number 1 to the carbon number 4 in our 1, 4s, and the carbon number 1 to the carbon number 6 in our 1, 6s. Okay, so we talked about some of the enzymes that we need. All right, so I got all my enzymes in orange here. We got our nice long one that we can get to in a minute. What was our first one that we talked about? We brought it up briefly in terms of breaking down that 1,4 linkage. Who has that in their notes from last time? Can you have that one? Glycogen phosphorylase. Here we go. So here comes glycogen phosphorylase floating along, and it says, oh, 1,4 linkages right at the end. Remember, it can only do the end units. So here it comes in, and it's going to take this guy, and it's going to break off this glucose right at the end. So then we have our free glucose, and we need to change that into glucose 1-phosphate. Okay? And in order to change that free glucose into our glucose 1-phosphate, this is where our hormone, or not our hormone, our enzyme hexokinase comes in. Okay? So let's go through that one more time quickly. We have our glycogen. Glycogen phosphorylase cleaves the end glucose off. Okay, so our 1,4 linkage is gone. And through the use of hexokinase, we now have our glucose 1-phosphate. Awesome. So we're going to stop there. What do we need to get that glucose 1-phosphate to eventually to get it into glycolysis? Not glucose 1-phosphate. Not G3, nope. It's a little bit higher up the chain. It's going to be our glucose 6-phosphate. So we got to rearrange that, and we're going to talk about the enzyme that does that a little bit later. But let's just stay where we are right now. Here we are. We have 2 nice branches, and we have our glycogen phosphorylase hanging out and being like, all right, let's do this. Let's break off some more end units. We got some end units right down here. We got some end units up here, but there's a problem. Glycogen phosphorylase only reduces glycogen 4 units away from a branch. It stops as soon as it says, oh, I know that a 1,6 linkage is coming up in 4 units. It says, that's it, I'm done. I can only break off these end units up to 4 units away. Question, Reno? No. Oh, technically. Just scratching the palm of your hand. Got it. So, glycogen phosphorylase hanging out has nothing to do now. All right. But we need to use this eventually to get these glucose molecules off. All right. So, you're on a construction team. Tristan, what are you going to do so that we can start using glycogen phosphorylase? Okay. Katie, what do you think? Add another glucose. Remember, we're trying to use glucose. It's a good idea. But how can we add another glucose with what we got here? Oh. Maybe I want to rearrange some of these glucoses a little bit so that I once again have at least 5 residues of glucose. So that's where a very aptly named enzyme, transferase, comes in. Makes sense. Transferase, we're going to transfer some glucose molecules. So what we can do here is transferase will look one away from our branch. It can't do the 1,6 linkage, but it can do this 1,4. And we can take this guy, and I wish I had a little bit more space, but we can kind of cram these guys in. And it can now make 1, 2, 3, 4, 5, 6, 7. Okay, you can't really see the one at the end, but now we have a chain of 7. So we have that guy all ready to go. But we got one more problem down here. We have our glucose that's on our 1,6. Well, we have one other enzyme, which is alpha 1,6 glucosidase, okay? But we're going to refer to it as the debranching enzyme, okay? We have transferase that moves the glucose from one branch to another, and now we have our debranching enzyme that once again can just debranch this 1,6 linkage. Boom. That guy's off. What do we do with this glucose now to get it into glycolysis? Who remembers from before? Bram? Oh, Ian? What do we need to turn it into first? I heard somebody. Glucose 1-phosphate, yep. So this glucose needs some hexokinase to be turned into our glucose 1-phosphate. Awesome. All right, let's see if I can kind of angle this guy so you can see them all. What's going to happen to this nice long line of glucose now? Which enzyme? Reno? Oh, the one we were using before. I remember that one. That one's a good one. Okay, who remembers over here? I heard rumblings. Allie? Glycogen phosphorylase. Remember, glycogen phosphorylase is our enzyme that takes them off when they're in a straight line. But remember, we can only do up to 4 away from a branch. But guess what? No branches left here. We got a nice straight line. We could do these, right? Take one at a time. There goes one glucose. Here goes another one. Here goes another one. Our glycogen phosphorylase is just having a field day in here, going one after another. So we've completely broken this up. Now we're left with a bunch of our glucose 1-phosphates. Okay? And let's remember what we did in order to get here. The first thing we did was we used our glycogen phosphorylase to take off any glucose units that were more than 5 units away, or more than 4 units away from our branch. Once we got to the point where we only had 4 units from a branch, we said, okay, we need 2 more enzymes. We needed our transferase to take any that were not part of that branch directly and move them to the end. And we needed our debranching enzyme to get that last glucose off. Once we had a nice straight line, glycogen phosphorylase came in again and started cleaving all of those because we had no more branches left. So we've been able to break down that branched enzyme into individual glucose units that through the use of hexokinase became glucose 1-phosphate.
So
Right.
since it's unbranched, glycogen phosphorylase can break off more than 4 at a time.
It's not breaking off more than 4 at a time, it's breaking off 1 at a time, but it doesn't have to stop 4 from a branch. That's why glycogen phosphorylase had to stop in the first place, because it saw, oh, four bonds from here, I know there's going to be a branch, I can't work anymore. Okay? Yep.
Do you know how it recognizes that it's 4 away?
Yeah, it used to date the 1/6 linkage, and it's like, you know how you get that like spider sense when you know it's like, I think they're here, I better get out of here. No, it's because of the size. Okay, so when we're saying it breaks down that one end link, it's not because, so here we have our 2 units like that, and our, where does it go? Glycogen phosphorylase doesn't just act on that end one, it's a larger enzyme that encompasses more than 1 glucose unit, and it just kind of takes off that end unit. You know, it's like, think about having a handful of Pez, all right, or one of a sleeve of Pez, you put your whole handle around it, and then you use your thumb to just pop off the last one. That's how it works. Does that make sense? These are good questions. What else do you guys have? John?
What was that enzyme that breaks off the alpha 1,6?
Okay, so the enzyme that breaks the alpha 1,6 linkage, we're going to refer to as the debranching enzyme. That's what you need to know. You don't need to know that it's the alpha 1,6 glycolysis, or what did I have? Glucosidase.
Is there a limit to how many glucose transferase can move from one branch to another? Or can it do like a whole chain of stick one?
I don't know the answer to that. For now, we're going to just say it's going to move the whole end of that on. I'm not sure if there's a limit, if it has to do like groups of 3 or something like that, but that's not really central to what we need to know. All we need to know is that it's going to take everything except for that direct branching glucose. Reno, did you have one?
Is the glucose 1-phosphate the same as the glucose 6-phosphate?
No. Glucose 1-phosphate is not the same as glucose 6-phosphate. That's a fantastic transition. Thank you for being a plant for me today. In the business, we call that a segue. Okay? So now we're back at all of our glucose 1-phosphates. That's what we have now. And we have to get it to be glucose 6-phosphate in order to get it into the glycolysis. So what are we going to do? So we have this. Where'd my glucose 6-phosphate? Oh, here it is. Here it is. Glucose 6-phosphate can go directly into glycolysis. Fantastic. We just need to basically move one of the side chains off that glucose in order to get it to be glucose 6-phosphate. So we need to move that phosphate from the number 1 carbon to the number 6. That makes sense, right? We are going to do that with phosphoglucomutase. Makes sense. Phospho is what we're moving. We're moving that phosphate group, okay? On a glucose molecule, we're going to mutate it. When you think about the word mutate, you think change. So we're changing where that phosphate group is. All right? So with the use of so we don't have those together. We have our glucose 1-phosphate, phosphoglucomutase comes along, and then after that, we have our glucose 6-phosphate, which then can go right through glycolysis.
So hexakinase turns glucose into G1P, not
Yes.
G6P?
Correct.
Even though it's
We simplified it when I talked about it when we did glycolysis the first time, and I said you need hexokinase, because that's one of the enzymes that you need.
Okay.
But we see now that we need a total of what? 1. Let's bring all of our star players out here. First, we needed our glycogen phosphorylase. Then we needed our hexokinase. We also needed our transferase. We talked about the use of our alpha 1,6, and last, our phosphoglucomutase. Quinn.
I was just wondering how you get rid of the alpha 1,6, like, because eventually you're going to be left with whatever the 4 glucose and the 1 alpha 1,6 link. So how do you get rid of all of it? Like
That's the debranching enzyme. The debranching enzyme is what takes that off.
oh, it breaks the 1,6.
Right. So let's go back there quickly. Good question. So remember, we got our 1,4s through here, and we had our glucose that was off here on our 1,6. Our debranching enzyme, we've already taken the other glucoses that were off here using our transferase. Remember, we moved those guys up here, but our alpha 1,6 glucosidase, or our debranching enzyme, is what takes this guy off. So that 1,6 bond between 2 glucoses is now gone. And then our hexokinase is going to turn these individual glucose into our glucose 1-phosphate.
So then the debranching enzyme breaks the 1,6 bond, and then the transferase moves it up to that upper.
No.
No.
Ah, fantastic. Okay. Our glucose 1,6, or our debranching enzyme, only works on that last glucose that's hanging on. Okay? It's not going to move this glucose up to the other chain. The transferase takes all the other ones that are off here and moves those up before our debranching enzyme works. So debranching enzyme only takes off one. If I was to make a synonym, it's very similar to our glycogen phosphorylase, but the difference is our debranching enzyme works on the 1,6 bonds, where our glycogen phosphorylase works on the end unit of a 1,4.
So then that one's just floating around before all the other ones start floating around?
Yes.
So hexokinase.
And that's when hexokinase comes in.
That's when hexokinase comes in and makes it into glucose 1-phosphate. Does that answer your question, Jonathan?
I have a question.
Oh, okay. Relax it.
Does the debranching enzyme, it only happens after all those other ones got cleared? It can't break the 1,6
Right.
if there's...
Great question. Okay, let's build that. Let's build that. So here's our top chain that we're eventually going to move to, and we have our bottom chain. Oh, no, we use our green ones. So right now, I know that my debranching enzyme acts on this 1,6. I can't come in here and break off these three. That's not how the debranching enzyme works. The debranching enzyme will only work after transferase has taken the end units off and put them onto the top chain. We'll only use our debranching enzyme on this guy right here. And that's really similar, once again, to our glycogen phosphorylase, because if we had a long chain, our glycogen phosphorylase, remember, can only break off this end guy. It can't come in and say, oh, I'm glycogen phosphorylase, I work on all alpha 1, 4 bonds, and break whichever one it wants. Remember, it's only popping off that last one. Last thing in the sleeve of Pez. Okay, which is really a good analogy. I'm really proud of myself for thinking that. Because what's Pez made out of? Sugar. All right. See, you went deeper with that one. All right. I've been thinking of that one for months.
I like your girlfriend one.
You like that one too? Yeah. Okay, so that's glycogen breakdown. That's how we get from this really nice. Let's go back to this guy now, our paper. That's how we get from this really nice kind of branch structure all the way to individual glucose 6-phosphate units. All right, so let's go through the list of enzymes in the order of use one more time. I want to make sure that we got this down. And Taylor, I'm going to have you start.
Yeah.
Okay? So if I have glycogen, what's the first? I'm trying to make some glucose units. I got a bunch of long chains, way more than 5 long away from a alpha 1,6. What's the first enzyme I'm going to use? Glycogen phosphorylase. Awesome. Okay. Brandon, you're up. Number 2. So now we have gotten to the point we only have 4 units away from a bunch of branches. Okay, so what enzyme do I need now?
It's not the transferase yet, is it?
No, it is.
It is?
Yep. Transferase. Okay, we have a bunch of groups that are 4 units away from a branch, from a 1,6 linkage. So we're going to take one of those branches, everything except for the branching glucose, and move it up. Okay, we've done that. Now, Emily, what's our next enzyme that we need? Not quite there yet. I can understand, hexokinase, but we haven't really broken off any individual glucose.
Oh, is that the debranching?
We need the debranching enzyme. So the debranching enzyme first, and now the hexokinase can come in. Yep.
Does hexokinase come in switch in from glucose to glucose phosphate one every time one's broken off, or does it wait till they're all broken?
Nope, it's always there. These enzymes are not kind of like hanging out in the bullpen, like waiting for something. They're all there all the time. Okay, and the right conditions need to be evident for them to work. Okay. Next, what do we got? What's our last enzyme?
Then we go back to glycogen phosphorylase.
Glycogen phosphorylase, because we got that nice big long chain now. All right, and we can individually chop up all the rest of those, and we have all of our nice glucose units, which through hexokinase are on to glucose 1-phosphate. Last step. Let's go, Dalton. What do you got?
So then the phosphoglucoamutase
Yes.
acts on the glucose 1-phosphate,
Perfect. And turns it into
turns it into glucose 6.
glucose 6-phosphate. Fantastic. You guys have just done it. You've got through all the steps of getting from a nice giant molecule of glycogen to these really very convenient subunits that can go right through glycolysis and make some ATP. That's the goal. Awesome. So we talked about that. Mm-hmm. Which part of the chains? Yeah. Glycogen phosphorylase first is going to break the end units at those alpha 1,4, but only up to 4 subunits or residues away from a 1,6 bond. Okay? Our transferase is going to be moving groups of glucose that are near a branch. Once again, up to that last glucose that is part of the branch. Okay? So the glucose that has the 1,6 bond in it, transferase does not act on. Everything else is going to move it. Our debranching enzyme works on those 1,6s. So right there at the 1,6, that's where our debranching enzyme takes that last one away. Okay? Our hexokinase takes our glucose and turns it into our glucose 1-phosphate. Our glycogen, nope, not that one, not that one. Our phosphoglucomutase takes our glucose 1-phosphate and turns it into our glucose 6-phosphate.
And how does it move the phosphates around again?
And mutates it.
Mutates it? Oh, I just mean like, where does it go to go from 1 to 6?
It goes from the first carbon to the sixth carbon.
Okay, so
Yep.
So, glycogen phosphorylate takes 1-phosphate and uses and turns that into phosphoglucomutase?
Nope. Phosphoglucomutase turns glucose 1-phosphate into glucose 6-phosphate. Okay. This video will be posted online. I recommend that you guys view it afterwards if you have more questions. A really great way to study would be to watch the video and write out the steps individually, okay? There may be a fill-in-the-blank type question similar to the last exam on the next exam. Hint, hint, hint. Okay. Where am I in my steps? That one, that one, that one. All right. Perfect. We got 20 minutes left. Let's talk a little bit about how glycolysis is regulated. We have a couple steps there. Right. Let's see if I can zoom. Oh my gosh. I'm like a technical wizard. All right. How is glycolysis regulated? First, let's talk about glycogen breakdown. Think of a hormone that may encourage you to break down glycogen. Ooh, who's that? Hunter, is that you? Is that you, Alex? Not norepi. Not insulin. Insulin's gonna make you store it more. Remember? When do you need a lot of energy? Yeah, adrenaline or epinephrine. You're close with norm, okay? So we fight or flight. You're running away from a bear. You have this adrenaline response, and you're going to be breaking down a lot of glycogen. So glycogen breakdown, adrenaline, epinephrine, increases phosphorylase's activity. All right, our glycogen phosphorylase, the expression or the amount of that waiting to act on our glycogen is going to go way up. So we're going to be popping the ends up of those PEZs way quicker when we have some epinephrine. Okay. Has anybody ever here had an adrenaline rush in a sporting event? Kimmy, can you recount that experience?
Yeah, it just feels like your heart rate picks up and then you just get like, I don't know.
Super energized.
Yeah.
Absolutely. I'm sure that many of us in a sporting event have had that happening. I remember that happening a lot when I wrestled in high school. Like you're about to get pinned and like, blah. All right, here I go. I'm getting out of this. Okay? So remember that epinephrine specifically is going to upregulate our phosphorylase. All right? Let's go to glycolysis now. So now to glycolysis. Couple things. How can we increase the rate of that? What do you guys think? Increase the amount of substrate. Okay, so we have more glucose, but what do we know about glycolysis? What kind of slows it down? What's that? PFK are rate-limiting enzymes. So we can increase glycolysis by increasing our rate-limiting enzymes. You could have all the glucose in the world, but if you don't have enough of your rate-limiting enzymes, you're not making any ATP. Oh, should I zoom a little bit more here, guys? Okay, that's one way. We can also increase our levels of fructose 1,6-diphosphate. We haven't covered that too much, but it's just a good one to know. And our last one is our concentration of oxygen. Why does that last one make sense?
Nothing happens as far as glucose is formed without oxygen.
No, that's completely wrong. Okay, but that's good. It's a fantastic answer. That's 100% wrong. All right. The answer there was, why do we need oxygen? Because nothing happens in glycolysis without oxygen. Well, that's not true because we know that this is anaerobic. But
does it have to do with producing lactate instead of energy?
yes. So Derek says it's because when you don't have oxygen, you produce lactate, and that's another thing that kind of gums up the works. All right? If we have oxygen, we're sucking that pyruvate out of there as fast as we can. All right? So you think about it, if we're making a lot of lactate, that's like putting your finger at the bottom of the bucket that's having water come out of it. Right? You get a hole at the bottom. But when we have oxygen, you're taking that out, and your end products, your pyruvate's moving really quick out of there, so you can break that down at a faster rate.
How does lactate slow down glycolysis? Does it interact with other
Just because it's a large amount of your end product being made. Okay, there is a chemical reason that I don't quite know, but think of it as having a lot of end product there. If you're chopping wood, all right, and you have a big pile over here, as that pile gets bigger and bigger on your outside, you're going to have to take more time to figure out where you can put that wood. Did you have a question? No? Good? So those are some ways that we're going to regulate glycolysis. Let's talk about one more factor for the Cori cycle, okay, and our lactate shuttling. We're going to zoom out here now. Excellent. So we talked about this before. We talked about lactate shuttling in our Lecture in the Dark series that we did. And we talked about the ways that you increase lactate shuttling. Remember, we increase our number of transporters that get that lactate out of the muscle, and we also increase our concentration of the enzymes that break that lactate down and turn it back into pyruvate. Okay? What is that enzyme? Hunter, what did you say? LDH, lactate dehydrogenase, okay? And we got increases of those MCT transporters that suck that lactate right out of the muscles that are being used and creating a lot of lactate, okay? And we said before, okay, we're taking this lactate and we're going to use our super Cori cycle to regenerate it over in the liver. But that's not the only place that you shuttle your lactate. You also are going to shuttle it to other muscles. Okay? So I'm riding my bike up a hill, creating a lot of lactic acid in my quadriceps as I'm riding up that hill. I'm getting that out of there because I'm well trained. I have a lot of MCT transporters. I have a lot of lactate dehydrogenase, but I'm still creating so much lactic acid that it's starting to accumulate. I'm shuttling as much as I can to the liver for regeneration through the Cori cycle, but I'm also going to shunt some of that lactate to other muscles. Marcus, where am I going to shunt that? What muscle is this? This is my quad. What muscle is this? What, your hamstring? Okay. Who has another idea? Oh. Scarlett says the bicep. Why? Not really using your arms. We have a lot of capacity to kind of oxidize and turn that back into pyruvate over here because it's a muscle that's not creating a lot of lactate. So we're creating a ton of lactate up here, so much so that we have to ship it off to other factories to remake it into pyruvate. So if this is our quad, this is our bicep, and our lactate is then going to get regenerated into pyruvate and get brought back to our working muscle there. Gwen?
Is that when, like, if you're sprinting, your arms get really tired, or because you're like moving fast?
No, it's because running and full pace running uses all muscles at the same time. So you're contracting your upper body muscles while you're running. It's not because you're getting lactate set there. No, but that's a good question. And that's another reason why swimmers create a lot of lactate, because they're using pretty much every muscle in their body. And another reason, another sport that uses a lot is going to be cross-country skiing. If you think about the cross-country skiing action, it's whole body. It's like this. And some of the highest VO2 maxes that have been measured have been in cross-country skiers. We've had some in our lab here. And one of our graduate students, Kyle, who's a coach, I think you guys have him for a lab. He just was in Kazakhstan doing some coaching internationally. And on our University of Wyoming cross-country ski team, we had the 14th place female in the world. Top American. Pretty awesome. And we also had the 44th place male. So this is really big news for our team here. So if you see any cross-country team, definitely congratulate them on a really good showing at that international competition. But this brings me to the last point that I wanted to bring up. And we're not going to take notes, but I want to talk a little bit about why do we measure oxygen during exercise testing? Because you guys are going to be doing VO2 max tests in lab, correct? Is that what you guys did? Or submaximal tests maybe first? So why do we measure oxygen? See how much our muscles are getting. Okay? Let's make it into physics. Let's make it into some math. That's what makes sense to my brain. Okay? Why do we measure oxygen? We measure how much oxygen goes in. We measure how much oxygen comes out. Derek?
Is it a limiting reactant?
Not a limiting reactant. We're talking about aerobic metabolism now. Okay? Because we're using oxygen. So we're doing endurance activities. We're not looking at force production. Okay? But what are we trying to determine? Why do we do a max test on somebody? Why would you want to do that? If we had two people, one person has a VO2 max of 50 mLs per kg, and another person has a VO2 max of 70 mLs per kg. These two people are going to race. They're going to do a 5K against each other. Who's going to win? 70. Okay. Why? Emily?
How efficient their body is working, like, so you would see how much oxygen you're using and expending to reach that highest point.
Okay. All true. Still doesn't answer my question. Now, those are all true statements. Yep, we're measuring it because we want to see how much oxygen they're using, but why? Why? Austin. It's not necessarily efficiency. If we're looking at efficiency, though, let's kind of build on that a little bit. What's our outcome measurement? You just correctly identified that a person with a VO2 max of 70 is going to win the race versus a person who has a VO2 max of 50. Why do they win the race? Able to create more ATP. What can you do with ATP? Let's go to physics. Work. You can do more work. And if you do more work in a shorter amount of time, which is power, okay, now we can do things faster. So we have these two people doing a race. We have a person who has a VO2 max of 70 versus a person who has a VO2 max of 50. We know the person who has a VO2 max of 70 is making ATP at a faster rate because that oxygen is the final electron receiver in our electron transport chain, right? So if we're consuming more oxygen, if we're using more oxygen, if we're making more CO2, that means that we're making ATP faster. If we're making ATP faster, that means we're able to do more work in the same amount of time. Okay? So if you have somebody that's making $100 versus somebody that's making $70, a person who's making $100 an hour is going to be able to buy more things quickly. If you have a person who's making more ATP in the same amount of time as somebody else because they're using more oxygen, all right, that's what we measure, they're going to be able to do more work. So the equation that we have there is if we're trying to convert things, we know that for every 1 liter of oxygen that you guys consume at any time, you are able to expend 5 kilocalories of energy. Okay? So if I consume 1 liter in a minute and Kenneth consumes 1 liter in 30 seconds, that means he's going to be doing work at a lot faster rate, right? Because he's consuming that oxygen faster because he's using more oxygen to get ATP. So what would be the difference? Why would Kenneth be able to consume oxygen at a faster rate than me? I'm breathing as fast as I can. That's not the limiting factor. Heart's ability to pump, okay. So there are a couple of factors there. So our ability to get that oxygen to the muscles in the first place, but that's not a limiting factor either. Derek?
His glycolysis is more efficient.
His glycolysis, so he might have an upregulation of some of his aerobic enzymes. Nope, we both have, we're exercising at a very high rate right now. We have about 99% saturation of hemoglobin. And we're going to talk about the hemoglobin dissociation curve a little bit later. It's one of my favorite lectures.
I'm just.
Oh, you're just resting.
Yeah, sorry.
Okay, if we're thinking about parts of a cell, what part of cells does Kenneth probably have more if he's able to do it? More mitochondria. Okay? Because we use that mitochondria in the electron transport chain. Alright? This is not going to be part of the test, but I wanted to kind of go through this because you're doing all these great measurements upstairs, but I want you guys to understand why you're doing the measurement. Why do we measure oxygen? And the reason we measure oxygen is because that's our fuel. If we don't have oxygen, we're not able to do any work. Okay? And a person who uses oxygen faster is able to do more work. It's basically like if you had 2 cars. All right. So if I got a Mustang and I got a Chevy Cruze, okay, a very responsible automobile, very good gas mileage. I got a Mustang and my Cruze. Which one is going to be able to do the quarter mile faster? The Mustang. Okay. In that quarter mile, which one's going to burn more fuel? The Mustang. That's like if you have 2 runners. The person who's covering that distance faster is utilizing more oxygen, okay, at a faster rate. Cool? All right, guys. So glycogen breakdown today, a little bit about lactate shuttling, and a little bit about glycolysis regulation.